Hello and welcome to our single-cell multiome analysis series with SciDAP. In this video we will be reviewing the output of cell ranger count that we've started in the previous video. You could see that it is done running, so now you can click on the sample and see all the statistics about this run. There are separate sections for transcriptomic reads and for ATAC-Seq. All of these QC metrics could give you an idea whether your experiment was successful. How many reads were actually sequenced, barcode matching, percent of duplicates. For gene expression, you could see it's about 73% here, which is pretty high. There's also more important information of the fraction of transcriptomic reads in cells. 85% is quite good. Distribution of reads mapped to various regions. Then in ATAC-Seq part, there are metrics such as a fraction of high quality fragments in cells, higher the better. So this is a very compact view of the most essential metrics, but if you'd like to see the full report from Cell Ranger, you could access it here. The most important metrics are on the top. Your estimated number of cells, ATAC fragments, and median genes per cell for gene expression. Provides you with some nice plots where you could see how much actual cells out of the total events were detected. Some nice Disney plots for ATAC and RNA. And my favorite, visual representation of how fragments are distributed among the clusters. There you could explore QC metrics separately for ATAC and RNA, which give you statistics per cell, and visualize TSS enrichment and read insert size distribution. This shape is what we would normally expect for good quality library prep. And for transcriptomic part, instead of transcription start site enrichment, we could find a table with genes differentially expressed between the t-SNE clusters. Now, when we're done with evaluating the quality of our sample, we could go back and look at the UCSC cell browser. There is an overview for RNA and ATAC parts. It means that the fragments will be named as genes or chromosome coordinates. This is the classic view of t-SNE plot in the UCSC browser. If you'd like, you could change it to UMAP here in Layout. If you look on every cluster here, you could see the differential genes for every cluster. Sometimes this is just more convenient than looking at the table. Additionally, if you don't like these colors, you can change the color palette to any other option from here. Also, for your convenience, you could change the circle size and transparency. Then, we can look at the genes of interest here in the Feature tab. For example, let's look at the interferon gamma expression. And it shows how the expression is distributed between the clusters. You could also change it to a different color. There is another interface where you could look at the average expression of the selected genes in a different way. Let's try another gene. You could see all the information here, percentage of cell expressing the selected gene, and what is the average expression for it. You could also split screen and search for different gene of interest on each side to compare the distribution of the cells that express this gene on your UMAP. If you know barcodes for the cells that are specifically interesting for you, you could find them using this tool, or you can find them with their IDs. You just provide the barcodes or copy-paste them from the list of barcodes that you might have from earlier. And you could also show only selected to have a better view on the particular cluster or a selection of cells that you need. So this was the quick review of the UCSC browser functions that might be useful at this stage. We will return to the UCSC browser later after filtering and aggregating. Finally, if you would like to download any of the files that were generated during this analysis, you could open the Files tab and find all of them along with the raw input data. Additionally, you could download the Loom file, which could be opened with the 10x Genomics Loom browser. In the next video, I will show you how to aggregate your datasets into one single feature barcode matrix using Cell Ranger aggregate pipeline.